So good morning, everyone, from wherever you're joining us. My name is Lewis Washira from Bioline Agrosciences. So I'll go straight to introducing our first speaker of the day. That would be Dr. Danny. Danny is a senior scientist with the International Institute of Tropical Agriculture and has spent over 3 decades working at the intersection of soil health, crop systems, and plant parasitic nematodes across tropical agriculture. His work has taken him across a wide range of African cropping systems, including long cycle and intensive production environments where nematodes are often present but not always recognized as the primary constraint. Dr. Coyne brings a rare combination of deep research experience and extensive field exposure. He has supervised 19 PhD students, published extensively, and played a major role in building nematology capacity across the region. Today, Danny will help ground our discussion. He'll walk us through the key nematode groups, the species that matter the most, how their damage typically presents in crops, and what growers and agronomists should be looking out for.
Thank you very much, Lewis. And wherever you are in the world, good morning from Kenya, which is where I'm based. And as Lewis says, I've got quite a visual presentation this morning. The ask to me was to provide some background and greater awareness and understanding of plant parasitic nematode problems, plant pests, and create more awareness to the audience. And so, especially with a general audience such as today, what I often do is provide an array of visual exposure to the symptoms, the problems, and the magnitude of the issues without dazzling you with data or numbers or graphs and whatnot. Colin will go into more detail into nematode interactions after I do, but mine is to set the scene and demonstrate what kind of problems do occur or you could expect on your farm. So I'm just going to share. Nice to see so many people registering and online, actually. I didn't think nematology was such an attraction, so it's nice to see it is. Right, okay. Lewis, is that up and running and in presentation mode?
Yes, we can see your presentation. Thank you for sharing.
Right, okay. So, as I say, this is nice to see a visual immersion into nematology, primarily the tropics and with a focus on Africa in particular, with an element of East Africa focus as well, but it's not confined to any particular region at all, apart from the tropics and subtropics by and large. Nice to see. So just initially, so I'm based in Kenya, where we have our NEM Africa lab. It's a nematology unit that are the bosses of ISIPI and IITA pertinently understood that nematology was an overlooked. There was a gap in nematology research and support in the region. And rather than individually setting up nematology units, combined forces between the two international institutes in order to have a critical mass, if you will, towards focusing on nematode problems. Despite the magnitude of the problem in the area, there was very limited nematology recognition. So NEM Africa was about 10 years ago we were born, basically. It is led by myself and by Solvig, respectively from each institute, and we act and operate as a single unit, which is quite unique, especially in the international world. And as you can see, we've got a dynamic group of young, energetic, youthful and dynamic team. And as you can also see, I bring the average age down quite markedly. But we've got a lot of interest actually locally through the universities and institutes around. And as you can also see, we've got a bit of an issue, a gender issue. For some reason, nematology seems to attract females more than it does males. Anyway, all good. And we've got a great team, youthful team. Nice to see some. Go. Right, so what is the issue with nematodes and why is there such a mystery around nematodes and nematology? Why are they so neglected, ignored, overlooked? And why is there a lot of misunderstanding? So hopefully I'd like to try, together with Colin, try and demystify nematodes, nematode pests, on the one hand, and create a little bit more awareness to the magnitude of the problem and the issues that we face. Nice to see. One reason that there is a lot of mystery is there's very few nematologists in general. So we lag way behind entomology and pathology, and there are very few nematologists in general in order to preach the gospel, if you will. And the other reason is that they're underground, out of sight, out of mind, and overlooked and ignored effectively. So that is one of the issues. A little bit technical, let's run through the nematology spectrum and give you an understanding of the overview of nematode types. This isn't particularly scientific, but for general jargon, we have a whole range of different nematode types, if you will. Plant parasitic nematodes, in fact, are a very small component of nematodes in general. The vast proportion of nematodes are actually free-living, beneficial, that live in the soil, marine environment, and freshwater environments. But effectively, every living organism has its parasites, whether it be insect, animal, slug, mollusk, fish, whatever. They all have parasitic nematodes. But today, we're going to focus on plant parasitic nematodes and the damage that they cause and the economic impact that they have on farming. Nice to see. Nematode is vermiform, worm-like. The key aspect here is the stylet. This is its tool, which it uses to injure plant tissue and to suck up plant tissue, food basically from the plant tissue. It uses a stylet to puncture plant tissue and cause the damage that we experience. That is the key issue here. Nice to see, however, despite them being worm-like, are entirely unrelated to nematodes. Earthworms are good, as are beneficial nematodes, but they are not to be confused with nematodes. Nice to see. This is what a group of infective early stage nematodes looks like under the microscope. All nematodes are microscopic, or the vast majority are microscopic, and all plant parasitic nematodes are microscopic. This is what they look like when magnified under the microscope. But as with all walks of life, they come in a whole range of sizes and shapes, macroscopic sizes, but a lot of different shapes and types. Nice to see. When we're working with farmers and we meet farmers and growers and managers, especially on a different crop, a comment we often receive is, "Oh goodness, we didn't realize that nematodes were a problem on flowers, on potatoes, on cherries, on whatever. We only thought that nematodes were a problem on tomato, and often in the tropics on banana, are two crops that are often associated with nematode damage. But the reality is, and the fact is that nematodes affect and infect and cause damage to every single crop that we grow, whether it be food, fiber, or otherwise. They do affect all crops that we grow, to varying degrees, of course, and depending on where you are and what the situation is. To the magnitude of billions of dollars every year, a huge amount of crop loss, is due to nematode damage and nematode parasitism. Estimates vary, of course, and we don't really know the true values. What we do know is that they cause billions of dollars worth of, or pounds, or euros worth of damage to the cropping industry each year. Nice to see. Right, just a little bit of technical aspect to help you understand nematodes and nematology. And we base them on feeding types and the way they feed. So, for example, there are a group of nematodes, parasitic nematodes called ectoparasites. And here we see the nematode feeding on a root tip. This is a rice root tip. Externally, from the outside, ectoparasitic. They live in the soil, the environment, and they feed on the plant root from the outside, using the stylet to inject into the root to suck out the contents. They feed round about the root tip. This is a different type of nematode, different species of nematode. Ectoparasite feeding around the root tip, where the root tip cells tend to be softer and more nutritious, basically. Then we have endoparasites that live inside plant tissue. And this is a rice root and another rice root. And as you can see, we've got endoparasitic nematodes living within. They enter the root and they move up and down. They're migratory, feeding as they move through the root. And as you can see, if you've got a root full of nematodes like you have here, these are stained. They restrict the use and efficiency of those roots if they're full of nematodes as opposed to root tissue. So we have migratory endoparasites that are mobile and move. And then we also have sedentary endoparasites, such as root knot nematode, if you're familiar with them, or cyst nematodes. The initial feeding stage is an infective juvenile, or the initial stage is an infective juvenile that enters the root and wanders through the root tip to find a feeding site, and then stays static and sedentary and stays in the same exact location, feeding from the same cells for its entirety of its life cycle, which is 1 to 1.5 months. We also have foliar nematodes, which often growers are very unfamiliar with and not aware of, that affect the leaf area, photosynthetic area. And then stem nematodes that it's a big problem with the bulb industry in particular, causing damage to the bulbs on the one hand, but also infecting the bulbs and creating problems with phytosanitary or movement of planting material. And they also can affect the product, in this case flowers, deforming the crop. So in summary, we have different feeding types. We've got ectoparasitic, feed from the outside, endoparasitic, that feed inside, which are either migratory, move within the root, buffet style, if you will, or sedentary, who are sedentary, stay put, and they are food is brought to them, if you will. And then we have also foliar and stem. Again, this is not scientific exactly, it's just to give you an understanding that there are different types of nematodes that are based upon where and how they feed. Right, so over to some symptoms. Below ground, if your crop is a tuber crop, the farmer, the grower, sees the damage, although they may not necessarily understand what the damage is caused by. And as you can see here, this is root knot nematode, Meloidigyne species, causing damage to various tuber crops, which is very visibly damaged. And if you have crops like this, it's wasted effectively and not of any economic value whatsoever. And I have seen farmers, carrot farmers, who've had to dispose of their whole fields as a consequence of nematode damage. But it's not always the typical galling damage that you see. This is sweet potato damaged by root knot nematodes also. And this cracking is a direct response and result of root knot nematode feeding and parasitism. And a consequence of nematode damage, especially to tubers and to roots, is that it manifests into rotting. So farmers may see a rotted tuber and think that they have a problem with rot. Yes, they do have a problem with rot, but it actually is manifested as a consequence of nematode damage and nematode feeding. Now, if the crop is not a tuber crop, the roots can still be and are still damaged, but they remain below ground, and often the farmer, the grower, does not see the damage and does not relate the damage to the loss of crop. Not always. Depends on how well aware the farmer or the grower is, and that's why we're here today. So last crop was celery, and here just giving you a few examples of the magnitude of damage that you can see, the severity of damage. This is cowpea, black-eyed bean, this is lettuce, salad, and this is coffee. So as you can see, depending on the type of crop, can often depend on the type of galling damage that is experienced and seen. Also, it can depend on the nematode species. Just a little bit of a closer look to the root damage, and you can see that although you've got galling that ends the root tip, you also have you also have necrosis and rot roots, which again affects the efficiency of the root system. This is pineapple, where root knot nematode is quite a serious problem to pineapple production, causing clumping and swelling of the upper roots in particular. Gone from root knot nematodes to lesion nematode. This is not the best quality picture in the world, but it does enable me to demonstrate what we're talking about. So this is maize. Now this was from an experiment grown in pots in the greenhouse. And as you can see, there's a marked difference between the two plants, which are exactly the same age, exactly the same variety and grown in exactly the same conditions, except one was inoculated with Patylicus lesion nematodes and the other remained uninoculated. You can see the damage and the extent of root reduction that nematodes can have after about 6 weeks. That was 6 weeks to 8 weeks. Banana corn, farmers may see this, but they don't always recognize that this is a problem or an issue. They may peel it back, but they would probably still use this corn for planting. And therein lies one of the issues, is that nematodes are transmitted or dispersed through the use of infected planting material. It's a banana corn, and these are the roots from infected banana corn. White is unaffected, and the red and the brown is nematode damaged by lesion nematodes on the root system. Through to above ground symptoms, to go through a number of symptoms, such as yellowing, fluorosis, not severe. This may be as a result of nematode damage. It may be a result of fertilizer, a lack of fertilizer or nitrogen, or it may be a combination of factors. And most likely is a combination of factors, not just one factor. But nematodes do reduce root efficiency, nutrient uptake, nitrogen uptake, and can lead and do lead to fluorosis and yellowing of plants. It can be mild as an indication to the issue, or it can be quite severe. Again, it's not just nematodes alone that cause this damage, and it's a combination usually of various factors. Sometimes the damage is very visible, but again, farmers don't always recognize that it's a nematode problem. This is a direct consequence of nematode feeding on the roots, disabling the root system, if you will, lack of anchorage, and especially when the banana plant is got a large bunch, heavy bunch, particularly in a strong wind, they go over and cause significant damage to crop losses. And back to the roots that I showed earlier, this is the reason, the cause of why that happens. Fields, and just to demonstrate here, nematodes often occur in patches within a field. They occur erratically, more often in patches, and you see a patchy growth pattern within a field of where the nematodes are. Baby corn, this is here in Kenya, in a large-scale commercial practice, and the manager indicated that they thought he had a problem with the irrigation system. We had a look at the roots, and we discovered it was actually massively infected with nematodes and causing significant reduction of growth and germination, patchy growth, stunted growth. And just to highlight, is a different style of nematode damage. There's no galling here. This is a maize corn root with 3 female root knot nematodes attached to it, no galling caused, but they cause significant damage to the crop despite no obvious galling damage. Over to the flower market, and this is a farm here in East Africa that I visited to look at actually a foliar nematode, but this was pointed out to me as well. This is root knot nematode damage on polyanthus, which is, I was informed, of quite a major problem on the crop. And that patch you can see is almost entirely due to root knot nematode. Similarly with pineapple, as I mentioned earlier, major problem for pineapple production is root knot nematodes. And here we have a guava. This is actually Brazil. This is a major commercial plantation in Brazil, which I visited, and we went to see the damage that root knot nematode was causing to guava. And as you can see, you've got really quite chlorotic, stunted guava trees here. Major reduction in output from these orchards. Across the road, I saw this, and I mentioned to my host, I said, "Oh, have you had a fire? It looks like fire's gone through the orchard." He said, "No, Danny." He said, "That is just an advanced stage of the problem of root knot nematode damage." However, there's a slight disclaimer there, and Colin will take this up later. Root knot nematodes, and as you can see here, the root damage, this is a woody root system extensively damaged by root knot nematodes, Meloidogyne. But it's the combination with fusarium wilt that causes the severe fatality of the trees, and severe damage. It's that combination of the two that proves fatal. And then we have situations like this. This is a nice looking, what appears to be a healthy potato crop in Chile. We were taken to this field purely and simply because it had a major problem with potato cyst nematodes. Yet when you look at it visibly above ground, you don't see any issue whatsoever. And often you don't see a problem with nematodes unless you have an infected versus an uninfected crop side by side. Back to the flower market, and apart from reducing head size of flowers, nematodes also have a major issue. There's a major issue with nematodes in that they create erratic harvesting dates, which interferes with the economic sustainability of the system. This is lesion nematode on chrysanthemums in Asia, actually. The stem nematodes that I mentioned earlier, they cause deformations, multiple tillering, stunting, etc. While the foliar nematodes reduce photosynthetic area and as well as crinkling and deforming the plant. This is a foliar nematode on strawberry. And just as an indication, this came from a colleague who works in Chile. And as a consequence of the rapid expansion of this nematode in the country, the government actually declared an agricultural emergency to it in order to address the issue. Foliar nematodes in Kenya, where we're based, we're starting to get some reports from growers, but not a lot. And I think there's probably more out there than we are aware of, and we do need feedback on that. And it's not just crops that are affected. Golf is a major source of income for some countries and some industries, of course. And turf has a major problem with nematodes, especially in the tropics. And if you like your golf and you pay lots of money to go and play golf, you want to have a nice green, not a patchy, chlorotic, brown patched green. And so a lot of money is actually spent on managing nematodes on golf turf as well as football fields, for example. Major issue. So we've seen some symptoms, but how important are they? We've seen some severe damage, and of course, damage occurs across a spectrum from slight or non-slight through to severe. I've shown you severe examples of damage, but please note that all these examples vast majority came from farmers' fields, actual real growing situations, and they were not from the greenhouse, apart from that maize one I showed you. So back to Chile, yes, they are important, so important that the government declared a national emergency because of a problem on strawberry, a key commodity for the industry in Chile. Closer to home, this was the headlines in the Daily Nation, the daily national newspaper here in Kenya 10 years ago, when we discovered potato cyst nematode. And I'm just going to run through a quick case study of a nematode problem on a key crop here in the region. So this was discovered 10 years ago as an invasive pest affecting the potato crop. It causes stunting, chlorosis, reduced growth, yield, reduced size of tubers, etc. And underground, this is what you can see under a heavily infested site. All those white little beads are female potato cyst nematodes, and close up you can see them all. They feed on the roots, suck out the contents, and reduce growth and yield. This is what they look like under the microscope. These are cyst nematodes, and each cyst encloses about 400 or 500 eggs, basically. That's one female that encloses it. And put it in perspective, this is a single cyst with a human hair by the side of it to demonstrate its size and difficulty in seeing them in the soil. In East Africa, we've now discovered that it's in at least 4 countries, and likely more. We're underway to survey other countries at the moment to see what the magnitude of the distribution is. But in an independent study by a socioeconomist, he came back with a figure of about $550 million annually is lost as a consequence of potato cyst nematode. Here in Kenya, a figure of around about $150 to $200 million is lost each year as a consequence of a single nematode on a single crop, which is quite extensive. What are we doing about it? We're doing a number of things, and one of which is to join with other colleagues to try and introduce resistant varieties that can overcome the problem. And here we worked with the James Hutton Institute, as well as many other partners, including the local government institutes and the Potato Institute, to introduce varieties. Last year, we claimed success in introducing 2 varieties that were very similar to the preferred varieties here, but with resistance to PCN, potato cyst nematode. And just sticking with potato, Irish potato is called here. And this is root knot nematode, and just to demonstrate damage on the impact it has on the industry. Institutes like KFC, Burger King, McDonald's, Lay's, who produce crisps and chips, will reject any potato with any sign of root knot nematode out of hand. It costs the industry massive. The local consumer, they still have to peel it more and lose more of the potato. But even when you have peeled it and cooked it, crisped it, or chipped it, it has a corkiness and it has a discoloring. And here you can see the nematodes, the female nematodes embedded in the tissue around the outer cortex. Major problem. So, are parasitic nematodes stealing your yield, affecting your quality, and reducing your income? Are you protecting your crops, your roots against nematodes? And what are you doing about it? Or are you even aware? Because at the end of the day, it's the roots that matter, by and large. There are foliar nematodes, but we're talking about roots here. And an infected root system is a compromised root system. Compromised root systems are inefficient. They're inefficient at taking up water, which requires more irrigation often, or poorer growth. It requires more fertilizer because the fertilizer that is applied is not accessed as easily and lost. Your production declines, as does your quality. And with a changing climate, hotter climate, and less water available climate, when you have roots, they're affected like this with nematodes, and nematode damage and reduction of the root system compared with a healthy system, there's a huge amount of loss to be had. But it's not just direct financial loss and yield. There's an environmental impact also. In the background, you can see nice, lush, natural forest here in East Africa. This is Tanzania, actually. And in the foreground, you can see the remnants of that forest. You can also see the remnants of the past potato crop. And there's holes that's just been harvested where this forest was removed in order to produce potatoes. Now, of course, deforestation occurs all over the world in order for agricultural practice. But if crops are underproducing and are not producing very well, farmers will open new land in order to have a better harvest and a better production. So deforestation is not necessarily purely down to nematode damage. Obviously, it's a combination of factors, but nematodes do contribute to that. And this is in Kenya, actually. Another forest has been deforested in order for potato production, which is a key commodity in that region. Management. How do we deal with nematodes? I'm not going to dwell large on this. I'm just going to spin over very quickly some of the management options available or possible. But key to all this is what we're doing exactly today is raising awareness as to nematodes, nematode problems, nematode damage, and the magnitude of the damage that they can do. And that involves talking to people, creating networks, enabling people to understand better why they should buy a resistant variety as opposed to a susceptible variety, for example, and capacity building, and that which we do at NemAfrica. And a lot of farmers say, do we have nematode problems? They're not sure. So is it nematodes or is it not? And this is where it requires sending material into a good recognized diagnostic clinic in order to establish whether nematodes are involved in the losses and the damage being experienced or not. Pesticides, we're all aware that pesticides, I put up a danger sign there. These are all nematicides that have now been removed from the market because they're so toxic. And one of the issues with nematicides is that they have to treat intractable problems that are in the soil in a large volume of soil, which requires large volumes of pesticides. There are good, better, more efficient, less mammalian toxic, environmentally damaging pesticides coming onto the market, synthetic chemical pesticides, as well as green products. And of course, EcoSpray partly involved in this today, and with the garlic NemGuard product, hits the green, more greener chemistries and safer products to use against nematodes in the soil environment. Healthy seed is critical, whether it be seedlings, whether it be vegetatively planted, propagated material, whether it be seeds, because nematodes are disseminated and transmitted through infected planting material, and it is a big issue for tuber and vegetatively propagated crops. This is potato from farmers' fields in Nigeria, and as you can see, heavily infected, and when used, just propagate the problem and the issue. Host plant resistance is a major cornerstone of nematode management, and as I mentioned earlier, we have been able to, together with everybody else, introduce resistant potato varieties into Kenya, resistant PCN, which I personally see as making a major difference to that $200 million or $500 million deficit that's come through. And there's a lot of interest in rotations and regenerative agriculture using cover crops and good rotations that will suppress nematode damage, as well as increase soil health for various reasons. And it's good to know what these practices are having on pest and disease populations, as well as soil health. Does it matter whether we control nematodes? Does it matter whether we work on nematodes? Well, in a changing world where we are definitely getting fewer nematicides, despite that one or two are coming onto the market now, losses will only increase. And the magnitude problem, particularly with the intensification of crop production practices, will create bigger losses from nematode damage and nematode infection and nematode damage. With that, I'd like to leave you with a happy smiling farmer face and hand back to Lewis to continue. Thank you very much for attending. Thank you very much for your attention. And I'm here basically. Over. Thank you. Lewis, over to you.
Thank you, Danny, for that. It was very informative. Guys, thank you for everyone who has continued to join. I'll do some housekeeping as we head straight to our next presenter. So first, I'd like to announce that this presentation is being recorded. So no need to worry. After the presentation, you will be able to get it on YouTube. I'm also sharing summaries of what the presenters continue to say on the chat. Feel free to share your questions in case you have any, and we will handle them at the end of the presentation. So without taking much further time, I will introduce our next speaker. That is Dr. Colin. I'll give you a bit of a brief. I'll give you what Dr. Colin is so that you understand what to expect from him. So Colin is a senior lecturer, that is Dr. Colin Fleming, is a senior lecturer and researcher at Queen's University, Belfast, with extensive experience in nematode biology, diagnostic, and applied plant pathology. He trained as a biologist at Queen's University, Belfast, and at the University of Montana, and has more recently been involved in teaching and research on plant parasitic nematodes in the Department of Plant Pathology at the University of Nebraska in the US. Givion has worked on plant pathology projects across much of Europe and previously led pest and pathogen diagnostics at the Department of Agriculture in North Ireland. Givion also served on the UK Plant Health Committee, supporting national plant health and quarantine quarantine decisions. In today's session, Givion will take us deeper into how nematodes function, from their biology and population dynamics to how they interact with plants and other pathogens, and then focus on a topic many growers struggle with: delivery and timing. So without taking much further, let me hand it over straight to Colin. Givion, it's a moment now.
Thank you very much indeed. Can everybody see that presentation now?
Givion, it looks clear.
Excellent. Yeah, great, thanks. Well, first of all, thanks to everybody for organizing this session. Really glad to be involved in the whole enterprise. And I'd like to thank Danny for the really great introduction to problems that nematodes cause in agriculture and horticulture. My background, as has been pointed out, I have over 40 years now involvement in nematodes. And I've been working on nematodes in agriculture, horticulture, forestry, and in sports turf. So I'll hopefully bring you some of my observations from those experiences over the years.
Yeah.
As Danny pointed out, understanding nematodes is key to being able to control them and manage them. And as he pointed out, the very first thing to understand is there are different types of nematode. They're not all the same. We've got the ectoparasites that are living on the outside of the roots, and we've got the endoparasites that are living inside the plant. A very important consideration is that plant parasitic nematodes are true parasites. It's not just simply, as with insects, an organism feeding directly on a plant. These nematodes are interacting at a cellular level with their host plant. So you can see in this slide, we've got the nematode. There's the nematode head at the top of the slide. We've got the nematode stylet or mouth part. And this is the plant cell. And you can see that around the stylet, there are changes occurring in the cytoplasm of the plant cell. It's becoming very active metabolically. But what is happening here is the nematode is injecting saliva into the plant cell and causing an increase in metabolism in that plant cell, which means that more and more food from the plant is being directed towards the nematode. So it's a true parasitism. It's not just simply a physical infection. The endoparasites bring this to a new height. We've got here a normal plant cell that you would see in a plant root. But look here where the female nematode here has been feeding. And you see the plant cells are changing size and structure, and again, becoming metabolically very active. So all of the nutrients are being diverted to the nematode from the plant root. And this really explains the dramatic effects we see in the plant upon infection by the nematode. Another important factor in controlling nematodes is understanding their life cycle. And the basic nematode life cycle consists of the egg stage, then 4 juvenile stages before you reach the adult. There are some variations in this, but this is usually the form that is taken by both nematodes. And the actual time it takes varies from 20 to 60 days to complete a life cycle. And quite often, temperature is the controlling factor in the time of this reproduction. So it's important to understand the life cycle before you can actually control the nematode. And I'll be addressing that in a bit more detail later on. The nematode life cycle, again, as I said, is fairly simple. Egg, 4 juvenile stages, and then the adult. In the case of the root knot nematodes, a very important factor is that only one of these stages is in the soil. The egg, the second stage juvenile, and then through the adult. Most of this occurs inside the plant infected root. Only the J2 stage is present in the soil. And this is important when it comes to controlling the nematode. If you're using a nematicide, this is the stage that is vulnerable. Inside the root, these nematode stages are often protected and less susceptible to chemical nematicides. So again, understanding the life cycle is really important. Danny mentioned lesion nematodes. These are really important in all areas of the world, from the temperate region up here in Ireland right down to Africa. And again, lesion nematode infection occurs very rapidly. And we're seeing, for example, in this case, nematode infects the root. And within 24 hours, we're seeing significant nematode damage in the root. Again, this is not just simply feeding damage. The nematodes are releasing chemicals that cause necrosis and cells to die in the root. And again, the impact you can see here on strawberries, where we've got a dramatic decrease in the amount of root. Uninfected plant on the left, and these 2 plants on the right are both infected by lesion nematodes. So very dramatic effect on the roots. And again, as emphasized by Danny, it's the root damage is, in most cases, the cause of the problem to the plant. As I mentioned, I've worked on a range of sectors: horticulture, agriculture, also sports turf. And here's a really good example of nematode damage in sports turf. Okay, so this is the football pitch at Old Trafford, where Manchester United play, and they had severe problems with root knot nematode. And this is basically what they were seeing. There were patches of turf where the roots were dramatically reduced in depth and in density, which meant that the turf surface was really unstable. And they've been combating root knot nematodes now for well over 10 years. So it's not just agriculture and horticulture. We're seeing the same things in sports turf, but the principles for control are exactly the same. You have to understand what nematode is present, what life stage it's at, before you can actually start to control it. So how do nematodes actually affect plants? But again, as Danny noted, one of the big effects is the effect on water uptake. If a nematode starts to attack a root system, we're seeing effects on water uptake between 1 and 3 days after the infection. This causes wilting, it causes decreased nutrient uptake, and it reduces chloroplast activity. So we're seeing a reduction in photosynthesis, and we're seeing a breakdown in proteins. The effects on photosynthesis are particularly important, as I said. In grassland pastures and turf, we're seeing a big reduction in photosynthesis 3 to 7 days after the infection by the nematode. What is happening here is the photosynthetic carbon is being diverted from the shoots of the plant down to the nematode feeding sites in the roots. So it's a big loss to the plant, and all this is being lost to the nematode, allowing the nematode to reproduce. Another big effect in plants is the impact on plant hormones. Nematodes, because they're actually disrupting the physiology of the plant and the metabolism in the plant cells, this leads to disruption in plant hormones. And we're seeing stunting, early leaf senescence, and a big decrease in nutrient concentration in the shoots. So a very big physiological impact caused by hormone imbalances. The actual effect on crops can be really significant. This is just some data from root knot nematode, Meloidicaila incognita, on rose cultivars, and we're seeing a big drop in root length, fresh weight, and the number of leaves. And as Danny noted, in flowers particularly, we're seeing a big impact on the harvestable flowers with regard to numbers and timing. So that's really important for producers. Another important factor to take into consideration is that some nematodes are actually transmitting plant viruses. So, for example, this is a nematode called stubby root nematode, parasitic. If you look inside the mouth parts of the nematode, these are virus particles. So the nematode actually infects plants with plant viruses, again, leading to additional problems. So if you're growing fruit particularly, that's a big problem when these particular types of nematode are present. So again, another consideration to be aware of. One of the most important considerations is the interaction between nematodes and other plant stresses. So this little diagram just highlights the relationship between the nematode population level and plant stress. And when I talk about plant stress, it can be anything. It can be drought, heat, disease, poor nutrition, saline water. These are all stresses that impact the plant. And you can see that if you've got low population levels and low stress, you don't get any symptoms. But if you increase the nematode level and increase the plant stress level, you start to see more severe symptoms. when you get high population levels of nematode and high plant stress, you can get very severe symptoms in your crop. So this interaction between stresses, abiotic and biotic, and the nematode levels is something that should be considered in any situation. This will actually result in decisions on whether or not to actually treat the crop with, for example, an expensive nematicide or put some other control measure in place. Now, this interaction of nematodes and other stresses is really well shown in the interaction with fungal pathogens. Root knot nematodes, during their infection process, will try to reduce the ability of the plant to combat against infection. And we have got this little diagram. And don't panic about this. This is just some biochemistry. We've got different plant defense systems. This is the ethylene system at the top, then jasmonic acid, and then the salicylic acid system. And these are all part of the plant's normal defense system. Root knot nematodes, when they infect the nematode, suppress these defense systems. Unfortunately, these plant defense systems also give defense against fungal attack. So if the root knot nematode reduces the ability of the plant to produce salicylic acid, that also impacts the plant's ability to resist fungal disease. And as Danny said, this interaction between, for example, fusarium and nematodes can have a big impact on the crop. Some of the effects we see when root knot nematodes infect the plant are systemic damage. That's the ability of the plant to take up water. This also weakens the plant's ability to fight pathogens. It reduces phenols and flavonoids. Again, these are essential defense compounds. So again, they reduce the resistance of the plant and they suppress the plant's immune system. In fact, what we see in some crops, we've got a resistant plant variety. When nematodes are present, that resistance to fungal pathogens is lost. So the nematodes are knocking out that ability of the plant to protect itself against fungi. So again, a very important consideration when controlling nematodes, be aware of other pathogens, other stresses that are present in the field. A really good example of this comes from here in Ireland. This is a golf green, and the greenkeeper here was having problems with persistent fusarium disease. He used every fungicide available, and he could not get rid of the fusarium. We found high levels of spiral nematode, and as soon as he controlled the numbers of spiral nematode, he was able to control the fusarium disease. So this is a really good example of an interaction between the nematode and another disease. And you've got to control both to allow the crop to have vigor. Now, controlling nematodes is a really difficult subject, and it's very much too much to actually discuss this morning. What we've got here are the various types of control measures that have been used over the years. Danny's touched on some of these: resistance, very important, rotation, and chemicals. Now, why are nematodes so difficult to control? Well, first of all, actually identifying the presence of the nematode is very difficult. You need to sample, and you need to have that sample analysed properly. When you do have identified your nematode, quite often nematodes such as root knot nematode will get a very wide host range, so rotation may not be practical, and it may not be giving you the required control of the nematode. The nematode body wall is selectively permeable. That means it is resistant to chemicals. And again, dormant eggs may be resistant to chemical control. And again, with the endoparasites, you're getting some degree of protection from nematicides being applied to the soil or the plant. And again, Danny noted that treating very large volumes of soil is very difficult and it can be expensive. When making a decision on whether or not to control a nematode, you have got to understand, again, what nematodes are present and what is the population level. This little diagram shows you the percentage yield delivered against the nematode numbers per plant. And you can see when nematode numbers are low, you're getting a fairly good yield. However, once you reach a certain point, and this is the tolerance level of the crop, you start to see a dramatic decrease in yield as nematode numbers increase. And this will accelerate to a maximum when after that you've got your maximum yield loss. And this could be zero depending on the nematode and the crop. So the relationship between yield loss and nematode numbers is quite often following this type of pattern. We've mentioned sampling for nematodes, understanding what nematodes are present in your soil. And you've got to understand that nematodes are not static in the soil, they can move. So for example, this is a sampling process that was taken on to look for dagger nematodes in soil. And you can see that the numbers of nematodes differ depending on the depth of soil. So if you sample just the first 10 centimeters of soil, you'll come up with a decision that there are small numbers of dagger nematodes present. But down to 15 centimeters, though, you can see the numbers are very much higher. So you've got to sample properly to actually detect the nematodes. And again, remember, nematodes can move through the soil. So if it's very warm or very dry, this nematode population may move deeper into the soil. And again, if you're sampling at a very shallow depth, you may not find the nematodes at all. So even diagnostic sampling is difficult and should be carried out with good instructions. This shows you different numbers and abundance of nematodes in some root soil taken from around coffee plants. And you can see the big differences in the numbers and the abundance of nematodes sampled during the wet season and the dry season. So again, timing of sampling is important. And quite often it's best to sample multiple times during the year to get an understanding of exactly which nematodes are present. For example, if we look at the wet season, we're seeing that the cyst nematode, Heterodora, very high numbers. However, if you sample in the dry season, low numbers are present. So you may get the false indication of what the important nematodes are present in the soil. So when should we actually try to control the nematodes? Well, the first consideration, what is the financial cost of the treatment? If it's going to be an expensive process, perhaps you only decide to treat high value crops. So that would be your fruit, flowers, non-staple type crops. And you would perhaps decide only to treat when the nematode population reaches a damaging level. So this is the damage threshold. After this level of nematode, you start to see yield loss. So was that your decision? You can see on the little table here with the different types of nematode and the damage threshold and potential damage. And you can see that the damage threshold differs depending on the type of nematode. It also differs depending on the type of crop. So, for example, dagger nematodes may have a very high threshold, damage threshold for one crop, a very low for a second crop. So again, you must understand the nematode and the crop you've got in the field. The other approach is to treat populations of nematode before they reach damaging levels. So you might decide, okay, I don't want my nematode population to cause me a problem in the future, so I treat immediately. So these are all management decisions that should be taken on the basis of the crop you're growing, the nematode present, and it's always good to get advice from an expert. So just a summary, for effective control of nematodes, you have to understand the nematodes that are present, know what the life stages are at various times of the year when you might be treating. You have to know the numbers of nematodes present to determine if they're going to be a problem. Be aware of any crops that are being grown or will be grown, and be aware of the soil conditions. Soil conditions are really important when it comes to, for example, applying nematicide. So very briefly, just to touch on chemical control and some of the considerations that you might be looking at. Danny mentioned the very large numbers of nematicides which have been used over the years and their extreme toxicity. And you can see here, way back to the 1860s, right through to the early 1900s and 1940s, 50s, we're seeing different types of nematode being invented. And you can see most of these are very, very dangerous and toxic. They're organophosphates, carbonates, and so on, fumants. And many of these have now been banned. You can see as we approach more recent times, again, some new nematodes are appearing, and the level of toxicity is reducing. Rather than danger, we're seeing the alert term caution. So they tend to be less toxic, but they're still to be used with extreme caution. So there are a number of nematicides available, but again, these can be expensive. There are different ways of actually applying nematicides. We've got direct soil application, we've got soil fumigation. You can apply in a granular form, so the nematicides are spread over the soil surface and just rotated into the soil profile. We've got broadcast application as opposed to row and band treatment. The latter, row and band treatment, you tend to concentrate the nematicide in the planting row, so that reduces the amount of chemical you use. You can also apply some of these in irrigation systems, drip irrigation or sprinklers. Again, for some of the nematicides, you can actually treat seed and root dip before planting. So there are different alternatives, and again, it's best to get advice from an expert as to which of these you would use. Now, to ensure good efficacy in using these chemicals, you must understand the soil conditions. The soil must be moist enough to facilitate the movement of the nematicide to reach the nematodes, but it shouldn't be waterlogged. You should try to make sure you're getting the nematicide in the soil profile at the correct depth. No point have it sitting on the surface if your nematodes are 50 centimeters deep. And again, if the nematodes are at the soil surface, you don't want your nematicide below them, deeper into the profile. It's best to use an integrated approach. So if you're going to use chemicals, also consider resistant crop varieties and rotation. This will all help reduce the overall nematode population level and the nematode pressure. Safety is always a big consideration. Some of these chemicals are very toxic. And again, best to get advice on their use. And again, timing is crucial. You've got to understand the nematodes present. You've got to understand the life cycle stage they're at in treating. So, for example, this is root knot nematodes. We've got 2 generations, egg to adult, egg to adult. And remember, with root knot nematodes, only the J2 stage is present in the soil. So that means this is the best time to treat the population. Apply your nematicide when the J2 stages are moving through the soil. If the nematodes are in the plant, they're less susceptible to the nematicide, and you may not get any control at all. So understanding your nematode and understanding the type of nematicide you're using is crucial to good control. And again, it all comes down to getting good advice and good management decisions. Now, all of you that are working with insects will understand the problem of insecticide resistance. So one of the big questions is nematicide resistance a problem? And the bottom line is it is, but it's not such a big problem as with insecticides. It tends to be a low field risk, but there is high potential. There are some cases confirmed in agriculture where nematicides have lost their ability to control the nematode population, and this is down to an increase and the ability of the nematode to combat the nematicide. This has been shown very well in laboratory studies, where we can take populations and work with them in the lab. But there's a different type of resistance, which is probably more important with nematicides, and that's the resistance in the soil. And this is caused by the breakdown of nematicides by soil microorganisms. And this seems to be the biggest problem. So if you've been using nematicides in a soil for multiple years, the soil microbes adapt to break down the nematicides, or using the nematicides as a food source, and this leads to accelerated degradation. We see it good in this graph here. This is a tomato field where tomatoes have been grown for 7 generations, and they've applied a carbamate nematicide annually. And you can see that the percentage control each year is dropping. This is not caused by the nematode becoming resistant to the nematicide. This is caused by the microbes in the soil increasingly breaking down that carbamate nematicide. So it means that by year 7, the nematicide is probably half as effective as it was in the first year. So this microbial breakdown is really important, where nematicides are being used year on year. And within the soil, it's things like Pseudomonas, Bacillus, Streptomyces, Trichoderma are all breaking down these nematicides. And this microbial degradation is affected by soil, pH, temperature, moisture, chemical structure, and in general, warmer, moist conditions tend to enhance this breakdown speed. So when it comes to using nematicides, you've got to be aware of the nematode, you've got to be aware of soil conditions, you've got to be aware of this potential problem of microbial breakdown. All of these are considerations that growers need to be aware of. And again, as I emphasize, you need to get good advice, first of all, on the diagnostics, and then on the actual management methods you can be using to control the nematode. So in general, if you want to avoid nematicide resistance, rotate your nematicide types as you would with insecticides. You've got to use the recommended dose rates. Dose rates have been produced for a reason. These are the rates that are known to be effective and will not be causing additional environmental problems. You have to use the recommended application numbers. So if it says in the label, use 3 times a season, don't use 4 applications. That again can lead to resistance. If possible, use resistant plant varieties as well and implement crop rotation. Not always available depending on the crop type and on the type of nematode. Again, some nematodes, such as root knot nematode, crop rotation is difficult because of such a wide host range. But all of these should be factors that you're putting into your consideration. And that's all I want to talk about this morning. So happy to hand back and I guess take any questions. If your microphone's on or off.
Yes. Thank you for the presentation, Colin. And I've been giving a summary of that on the chat. So in case you've not been able to follow, please just check the chat and you'll be able to have at least some notes on what has been happening. So for those who couldn't hear me earlier, I will do an introduction. It's a bit late to be doing the introduction, but at least to give you an idea of what we're doing here. So this is RootCamp, a webinar by Bioline Agrosciences. We are currently on our 4th episode. We've been doing this for the last 1 year and a half or so. And the idea is that farmers try to control many of the pests and challenges they face above the ground, but normally forget one key challenge that they can't see, which are nematodes below the ground. We started, he was able to tell us about nematodes, the kind of nematodes we would normally experience and the damage. It was quite insightful to learn that nematodes can get to a point where they become an emergency, as we saw the case in Chile. We then proceeded to Dr. Colin Fleming, all the way from Ireland, who went a level deeper into the biochemistry of the nematodes, how they feed and all that. So without taking much time, I want us to go straight into the Q&A session. So this is the right time for you to, in case you have any questions that you've not that have not been resolved in this presentation, or something that you're curious about, please feel free to share it in the comment section, the Q&A section of this meeting. So I'll start with our first question from Williams, who asked, what is the typical lifespan of a nematode? I've seen there's a bit of chat there between Kevin and others, but I'd like our expert to handle this. So between Danny and Colin, I'm not sure who will handle this. So what is the typical lifespan of a nematode? And probably a continuation of the same question. How many eggs can it lay during its entire lifespan? And how does Bacillomyces work on endoparasitic nematodes? And I think a fourth question, it's all about understanding nematodes. What is the most destructive stage? So
it depends on the nematode and the circumstances. And as Colin indicated quite clearly early on, is that temperature is very relevant to the lifespan. So a low temperature will lead to a longer lifespan or life cycle than a high temperature. So in the tropics, nematodes multiply much faster than in the temperate areas because they have a much shorter lifespan. And that's one reason why nematode problems are more acute in the tropics than they are in temperate areas. It depends on the nematode, it depends on the feed, it depends on numerous things. But a root knot nematode, which is the commonest and most problematic out of all of them effectively, is between 25 and 60 days, depending on the temperature, I think. And Colin can correct me there.
Yeah, that's correct. Again, some of the nematodes can last multiple years. Some of the larger ectoparasites may last 1 to 2 years, and they can reproduce multiple times during that, again, depending on temperature. So it's a lot of factors affect the lifespan and the numbers of eggs being produced. In the case of cyst nematodes and root knot nematodes, Danny, again, keep me right, we're talking hundreds of eggs being produced by a single female.
Yes, correct. And that's an interesting point, actually. I'm not entirely sure on root knot nematodes, but it's like 200 to 300, I think, per female. Again, depending on the circumstances and how well she is fed. There's all sorts of factors here, but on average. But cyst nematodes tend to be around about 400 or 500 eggs per cyst, per single female. But here in Kenya, where PCN, potato cyst nematode, has been introduced, we found that cyst size has actually been dramatically increased. We're not quite sure why, but we believe they have better conditions and feed better than in temperate climates, and we're getting up to 800 eggs per cyst. So interesting. Over.
Yeah.
So the only other element of that question is, what is the most destructive stage, and how does Bacillomyces work on it?
Yeah, that depends on the type of nematode. In the case of the lesion nematodes, which are migratory endoparasites, they cause damage by moving up and down the root system, causing, again, physical damage as well as this general metabolic damage. And it would be the juveniles and adults. In the case of cyst nematodes and root knot nematodes, the most damaging stage is the female, because she is the stage that's actively feeding and taking up most plant nutrient. So Danny, maybe you want to add to that?
Not really, Colin. Like you said, a lot of these questions are dependent upon a lot of different factors as well, really. But like you say, in some species don't have males actually. But in some species, the males do not feed, so they're not damaging at all. In other words, females are by far the more damaging gender.
Great. And for our next question, so how often should you be checking for nematodes, just to be sure that you're safe?
Again, it depends. Everything depends, and each individual situation is quite individual. And so this is where there's a lot of one-to-one or farmer to extension agents or extension service information service as to how best to add a sample, when to sample, what to sample, how to store, and where to send samples for assessment, basically. It's a little bit difficult to generalize, but as Colin pointed out, when it's raining, nematode densities tend to be higher. So that is better than when it's during the dry season or during the winter. So when they're most likely to be active and not necessarily, well, I was going to say not necessarily at the beginning, but if you want to have an assessment of your potential nematode damage and therefore plan as to whether you should apply a nematode management option, you sample before planting to assess the potential damage that nematodes may cause and therefore make a decision as to whether to treat or not. But if you want to find out what kind of densities are from a nematological perspective, then mid-season is usually better to establish how treatments are working, basically. So in a trial situation, from a researcher perspective, we would sample an annual crop, mid-season to maturity, flowering to maturity.
Great. And on that, still on the same, a different question from Ashok, but I think it's connected to that. It's what months would you recommend to apply nematicides? Like you mentioned, during rainy seasons, the populations tend to go higher. So how would you advise the control cycle to be?
Well, again, I wouldn't provide a generic application schedule. Everything's individual and depends. And applying nematicides has implications to the environment, to the crop, to everything basically. So I would advise that people seek local advice from agro input dealers or extension agents as to when best to apply, based upon the nematode situation, the crop, the season, the conditions, greenhouse or open field, for example, etc., etc.
So there's a question here from Washira who asks, and how does flooding work as a means of physical control, yet PPNs are water loving? What's the science behind this? I think as you answer maybe flooding, you can also cover solarization as another method.
Yeah, go ahead. I'll go ahead. Yeah. So basically, both using solarization and flooding are methods to reduce nematode levels by making their local environment hospitable for them. So solarization is probably the more common approach. And what you're trying to do is to increase the soil temperature. Nematodes will be more active as temperature increases, but at a certain point, a higher temperature will kill the nematode. So in the temperate regions, you find that the nematodes up here in Ireland, for example, once you hit 26, 27 degrees, start to struggle. In the tropics, the tolerance level of nematodes for temperature is much higher. So you've got to get higher temperatures, maybe 40s, 50s, Danny, I'm not sure, to actually kill the nematode. Flooding has been used in some regions to control nematodes. What you do is you flood the field, and again, you're trying to remove oxygen and the ability of the nematode to respire. And again, you're trying to reduce the nematodes. The problem with that is you can get other forms of damage. So it tends to be less used in solarization, which can be a quite effective means of control. And again, environmentally, solarization would be much safer than using nematicides, but there are other problems as well. Danny?
Yeah, not much to add really. You covered it pretty well there, Colin. But just add, like, flooding deoxygenates the situation which nematodes require. But then you may ask, well, actually, you pointed out rice as having a nematode problem earlier, and paddy rice does have a nematode problem, and I believe they derive their oxygen from the plant, so they can survive flooded conditions, but access oxygen from the plant. In terms of solarization, it can be very effective. It's not entirely practical on a large scale, but depending on the cost-benefit ratio and the value of the crop, it can be on large scale commercial basis be very effective. For good, effective solarization, the soil needs to be moist, so it's actually the water that heats up more efficiently, and then that's what kills the nematodes. Dry soil is nowhere near as effective as moist soil. The nematodes are then more active and more prone to being killed by temperature.
The other important point to make, Danny, is that parasitic nematodes are essentially aquatic organisms. They're living in the water film around the soil particles.
Yeah, correct. Yeah, which is what I think the questioner said. They're aquatic organisms, so why does flooding affect them? But yes, essentially they're aquatic organisms. On the solarization, this can be, certainly in my part of the world in Africa, it may not be practical on a large scale or basis, but on a small scale, it can be used very effectively to manage nematodes in a nursery situation. For example, if a farmer is producing seedlings for the transplanting to field, so you can effectively control the nematodes in the seedling nursery, and then they're transplanted as much as possible free of nematode damage and other fungal pathogens as well.
Yeah, interestingly, in the UK, in golf courses, in some football pitches, and at Wimbledon Tennis Club, they're using sheeting and high temperature steam injected into the soil profile to kill nematodes. And again, it's a similar process to the solarization, but using steam as the heat source.
Yeah, steam's used in some sort of glasshouse high value crop flower production as well. Actually, I've seen, but it is expensive.
Great. Thank you for covering the physical methods. Maybe just very quickly, mention about biological controls such as marigold and rapeseed before we head to our final question, which is about resistant management. Just maybe mention something about marigold and other plants that can be used to control.
Yeah, I've certainly used. Sorry, go ahead, Danny. Go ahead.
Carry on, Colin.
What I was going to say was I have used rapeseed very successfully in some situations as a pre-planting crop. Again, you basically mulch the rape or ground up seed into the soil profile. That releases isothiocyanate, I think, which will kill the nematodes. So it's basically like a biological fumigant, and it can be quite effective. But again, getting good levels of control can be difficult depending on the soil type and again the size of the field and the crop that's coming in next. Marigolds I've less experience with. I've seen that used, but again on a small scale, not really in a large agricultural field. Danny?
Yeah, this comes down to a host rotation, a host resistance, susceptibility, and rotations as well to some degree. The reference to rapeseed, I assume, is what Colin's just said, where you chop it up and you mulch in brassica crops, which have high levels of isothiocyanates, which are released and which kill the nematodes. It can be effective, but it has to be done properly, and different crops have different levels of isothiocyanates. So you need to select your variety or your crop quite well also. In terms of marigold is well known for being a non-host of root knot nematodes. Now, I'm not sure if it's certain species that it's resistant to and that perhaps some species it's susceptible to. But certainly in Southern Africa, it has been used on large-scale basis, and it used to be a component of tobacco farming where tobacco was very heavily damaged by root knot nematode problems. And by putting a rotation in with marigolds, it enabled to clear or reduce, rather, suppress nematode densities that made it profitable and cost effective to actually use marigold. There's a number of crops that are out there. Root knot nematodes are particularly damaging because they have such a wide host range. They're incredibly feed on a huge number, which is unusual for a parasite. And so they're very difficult to manage in general, and that's one reason why they're so problematic. Finding useful rotations is quite difficult. Resistance does exist, but on a generalistic crop basis, most crops will be affected by root knot nematode. Over.
Perfect. So let's tackle our final question before we call the meeting to a close, because we are almost at we're actually at closing time. So this is from Murray. He asks about how long is the resistance suppression to fungal attack once the nematode feeding begins? How long is the resistance suppression to fungal attack once the nematode feeding begins? And another question connected to that from Regan is, why is it important for growers to set a lower action threshold for the virus vectoring nematodes like Trichoderus? So let's tackle those two questions. I see there's still more questions that I've not been able to tackle, but we will get back to you after the webinar. But for now, as we close, let's tackle those two questions and then close the meeting.
Yes, just on
to Colin.
Yeah, I'll answer the first one on the fungal susceptibility. The root knot nematode, when it establishes a feeding site, it starts to knock out the plant defense. It's not a one-off event. The nematode has to maintain that feeding site. So it's continually injecting chemicals into the plant, which suppress the nematode's immune system. So it's an ongoing process. And as far as I know, that process will continue for the entire life cycle. So that loss of resistance to the fungus continues while the infection proceeds. Maybe Danny would like to take up the second question on the Trichoderus.
Yeah, I don't have a huge amount of experience with the virus transmission, actually. But when that question, what was the specific question?
It was about why growers should set a lower threshold, lower action threshold for virus vectoring nematodes such as Trichoderus. Why is it important for growers to set a lower action threshold for virus vectoring nematodes such as Trichoderus?
Well, we have quite a lot of Trichoderus and Paratrichoderus here, Trichodorids, which are quite problematic. Not sure to the extent that they're transmitting viruses here. There's been very limited, little, if none, research on the actual virus implication. Setting lower thresholds, I'm actually not sure what the context is here on that, and so I'm not going to shoot a question and shoot an answer that I'm not sure of the question actually, and the circumstances.
In general, I think, in general, it's the amount of damage that the nematode will cause. So if there's potentially for very high levels of damage, you have a lower threshold, an action threshold. If the nematode damage is less of a problem, you can cope with a higher damage threshold level. That's just a general consideration.
Yeah, so thank you for that. Without further ado, I will now call the meeting to a close. I'd like to give special appreciation to all the groups of people that have come together to put this. The speakers, that is Dr. Danny and Dr. Colin, the Bioline team, Lewis, John, Corey, Barnaba, Benjamin, and the entire team. Our sales team who have done a lot of mobilization on the ground, Keegan, Susan, Anne, Faith, Festus, Francis, Phyllis, Ismail, Grace, and all the others. The EcoSpray team led by Lee, Keegan, Regan, Peter, Mary, Ant, and of course, our audience. We have growers, we have students and professors from the field of academia. We have agronomists and all BioLine staff who have made time to attend this meeting. Thank you so much for your time. Thank you for your participation. If we didn't cover your question, we will take note, share it with our speakers, and eventually give you a response. In case you didn't catch the entire presentation, no worries. We are recording it, and you'll be able to access a recording of it on YouTube, probably in the coming days. Once again, thank you for attending. Remember, RootCamp is a continuous series. This was our 4th episode. We will be having more episodes covering different topics. We also have control over that. Feel free to reach out to us in case you feel there are pertinent issues that we should cover. And with that, I'll call the meeting to a close. Thank you and see you next time.